Hello, I am Larry Boise from the Winship Cancer Institute at Emory University, and along with my co-chair, Dr. Paola Neri from the University of Calgary, we are at the Myeloma 2022 in Scottsdale, Arizona, and we just finished up with a really exciting session on novel targets in myeloma. You know, Paola, I love novel target sessions because there's a lot of variety since there's so many different types of targets, whether they're precision medicine approaches like venetoclax that I talked about, whether they're immunotherapeutic approaches like CAR T cells or T cell engagers like you discussed as well. And we also heard about a new target, PIKfyve, and it's moving potentially into the clinic in the coming year or so. And so I've always loved to hear about that. And of course, we got to hear about some older ones too, even proteasome inhibitors and IMiDs, and learning more about potential resistance mechanisms too. So what was your thought?
Yeah, even RAS, the concept of targeting RAS directly, indirectly, in the past we thought was very difficult. Now we have a way to do that. So I think to your point, technology is allowing us to use different approaches to target things that we know in myeloma biology are very important. I may also comment on the fact that, even studying, using more the immunotherapy, for example, have allowed us to start understanding if we have predictor of response to this therapy that in the past we didn't think were possible. We start learning mechanism of resistance to these approaches as well. So I think analyzing the tumor as well the immune cells have allowed us to understand at different level really what matter for a patient to respond or not to an immunotherapy.
Yeah, and I think a great example of that was actually in the second part of your talk when you talked about the T cell engagers and what was involved in where you saw responses and non-responses. Can you tell us a little more about that?
That was interesting because we're always wondering if we have a way to really tell up front if the patient we're seeing clinic will be a good candidate for immunotherapy. And I think our initial data seems to suggest that unfortunately if we have dysfunctional T cells present in the bone marrow, in the peripheral blood, these patients have very they don't have much ability to respond to T cell engager. In contrast, the patient, they have healthy, for example, memory T cells in their peripheral blood, even if there is an exhaustion component in the bone marrow, this could be replaced by this healthy pool of T cells that by being exposed to cell engager could just come in the bone marrow and having an anti-tumor effect. Something very unique that I think argue about maybe the peripheral blood will be sufficient for this patient to be profiled up front and make the decision based on that.
Yeah, and I know that's going to be really important too. It's so much, I think, more convenient for the patients themselves to have that profiling done that way. And so while we're thinking about immunotherapy, you also talked about, with so much excitement now with our second FDA approval for a CAR T cell. Mm-hmm. However, even with this excitement and these great responses that we're seeing, there have been some issues with the duration of the response. And so you also talked about one potential way of armoring a CAR T cell to maybe get over some of those issues. You want to tell us about that? Yeah,
this is actually using one of your favorite genes, the one you studied many years ago, the BCL-XL, BCL2L1, that actually is a pro-survival gene, and we identify clearly was much higher in the CAR T from sensitive patients. So we have actually armored CAR T using this specific gene, thinking that we could overcome the activation-induced cell death, we could increase the survival of the CAR T in patient. And actually we have done for now in vitro and in vivo studies showing that indeed these CAR T armored are able to survive longer, to persist longer, and have longer also anti-tumor effect. The next step will be talking about translating into a clinical setting, thinking that with the catch there, and I think you can comment on that too, how how safe it is to introduce a pro-survival gene in a CAR construct. Do we need to be concerned about, safety or inducing other issues? What is your thought there, Larry?
No, I think it's something we will have to consider moving forward. I mean, we know that even BCL2 itself got its name because it was an oncogene, right, in follicular lymphoma. So introducing genes like that, there are going to be. We'll have to just keep a real careful, look at. Will they affect the T cells, and in a way to transform them could potentially be harmful to the patient? But I so far in your data, it does not appear that that's going to be the case, and I think that's really important. But we will have to monitor that moving forward.
And we could also, right now we can manipulate the product to even eventually add another gene like CD20 that can be turned off and on with, for example, rituximab. So I think we are living in a time where there are a lot of strategy that we can also use to improve the product, to improve a CAR T, to make T cells more memory-like, less exhausted, that we know matter for their response to patient. So I think in the next few years we'll be very excited to see this coming up with new discoveries.
Yeah, absolutely. Inducible suicide switches for the cells, all those would definitely be ways that can be tested. So one of the other parts about think about novel therapies, a lot of these we're talking about are really down the road for patients, but we still have our backbone therapies, our proteasome inhibitors and our IMiDs, and we're still learning so much about them, even though we've been using them now. We're getting on to almost 20 years of using these drugs, and I think we heard several talks, 2 talks today, really about that. About are there potential markers of resistance that could impact the responses to these backbone therapies. Dr. Orlowski discussed RAS mutations and how that may actually affect proteasome activity. And I thought that was a really interesting possibility. We'd all love to target RAS for sure, and we do now have a drug that targets RAS, but unfortunately, or for better or for worse, myeloma patients don't seem to have the mutation that drug targets. But I still think it's very important to understand how the genomics that we are learning about influences even our backbone therapies. I don't know if you had any thoughts about that.
Yeah, and actually, as a follow-up as well, I was very intrigued yesterday too. We heard about this immune effect of proteasome inhibitor, of the IMiDs that we kind of knew, but then we start learning why in combination API and IMiDs perform so well. They induce a lot of response in patients because again, there is an anti-tumor effect, but also an immunomodulation effect that is equally important, especially when we talk about combining them together in patients with myeloma. So I agree with you. We are learning a lot by even deeper analyzing the cause of resistance in our patient, because then we could combine them in a better way and eventually overcome any of the resistant mechanisms.
And we also heard about a really intriguing possibility that, again, I think we're still in early days of, is understanding how the genes that are involved in the proteasome or in the response to IMiDs, those genes themselves are being regulated. Martin Kortum talked about that. I don't know if you have any thoughts about that. Methylation,
epigenomics, I think is an aspect that we didn't actually explore until really recently. And we're learning that is extremely important to understand even that component, because many genes are on and off regulated by epigenomics events. So I think it's another, where we talk about what are the best methodology to understand this factor. I think we have to include epigenomics or methylomics as another way to study this aspect.
And not just understanding the biology of it, but also, are these going to be markers that you can use? Can you use methylated DNA or regions of methylated DNA as markers of sensitivity or resistance to these therapies? One of the other, again, exciting, I think, studies was a new drug, the PIKfyve inhibitor that Dr. Keith Stewart discussed moving forward with. And this was very interesting in that he actually first demonstrated this as a potential target actually a few years ago, and now has moved this forward where I think we could see clinical trials in the next couple of years.
Yeah, I talked about combination with iberdomide, for example, with selinexor. So these are other anti-myeloma drugs that, of course, we will be interested to see how do they work together, what is the toxicity profile that we see in our patients. So to your point, more discovery bring novel combination for our patient as well. And probably one more thought to just about, again, antigen escape, for example. We know which are the target expressed by the myeloma cells, but now we're learning that the cells overall have a way to escape that if we target only one element. I show you some cases with the BCMA loss. How important is now to have additional target, eventually have dual targeting approaches that in the event one of the target gets lost or downregulated, we have still a way to capture and to target the myeloma cells to induce killing. So I think even that aspect need to be further explored, but there are no doubt. I think in the next section will be interesting to see the clinical data for these immunotherapeutics.
Absolutely. We also, when we think about novel targets could be, there's so many potential targets that we know about, but we haven't been able to reach yet in myeloma. And one of the real potential opportunities there is through the use of these PROTAC drugs, which target different proteins for degradation, actually working very similar and based on how drugs like IMiDs work, like lenalidomide and pomalidomide. And we did have a talk from Dr. Shaji Kumar on this, showing a really what I think I would call it more of a proof of principle PROTAC, and targeting the same molecules that the IMiDs do, Icaros and Aelios, and he really showed some very exciting data with this. Again, I don't know that we are whether this will be a drug or not, but the idea that they worked very well, and it was much more potent than actually pomalidomide, at least in causing that degradation. So I think it opens an interesting opportunity. I'd love to see more on that. And are they also allowing for the targeting of? Is it just specific to those, or is it also affecting other molecules?
Yeah, so how specific it is, that I think it will be key to moving forward eventually for the clinical trial, see the specificities, see also in patient how easy it will be to deliver a PROTAC drug that maybe have some issue on transferring in patients. But I think to your point, we may this way target things that will never be possible before. So no doubt there would be a potential new approaches.
And so then finally, you talked about putting BCL2L1 into the CAR T cells to armor them so that they would be more persistent. I actually talked about the original, which was BCL2, and targeting that in myeloma. And there's been very exciting work actually done by people like Dr. Kumar showing that a subset of myelomas that have the T1114 translocation are very sensitive to this. And so we've been trying to understand, can we have better predictors of this? Because even within the T1114, it's only about half of the patients that respond. And so we've been working on different ways to do this. And we've been focusing on functional approaches to actually measure this. And I think so far the data is promising on that. Yeah,
and I'm actually curious because you comment also about the flow panel. What is the best way to even predict response in patients with 1114? Can you comment a bit more on what component with marker do you think need to be there to be able to tell that?
Sure, Paola. This was really based on work that we had published a little less than a year ago, trying to understand what were the factors that controlled venetoclax sensitivity in myeloma, and through several different genomic approaches and looking at gene expression in sensitive and resistant samples and cell lines and patient samples, we determined that it was a subset of myelomas of mostly 1114, but not exclusively, that express B cell marker, so a more immature cell than where a myeloma cell is. And we know that within the 1114, there's a group that was originally described transcriptionally as the CD2 subtype, and so it kind of fits with that group. There are surface markers that are expressed on B cells that are not normally on plasma cells, like CD20 and CD79A and CD79B and a few others. And so we actually, in that paper, developed a flow panel that could be used, because flow cytometry is used much more commonly in a clinical testing lab than some of the functional tests that we've been doing. And it may be a way to translate that. Study worked really well in that, so we're still working on it.
Yeah, from your perspective, having a B cell-like phenotype seems to be still a better predictor of venetoclax sensitivity. Why, when they become more plasma cell-like, and we have initial data on that regard too, they probably be a sign of less activity of venetoclax. So something, to your point, that we need to investigate further, because again, not all the 1114 respond to venetoclax. It would be nice to have a test that we can easily use and predict the response just based maybe a flow panel.
Yeah, and I think that's what we're shooting for. So we'll maybe at the next meeting, so in Myeloma 2023, we'll present more data on that as well. And so I think that really sums up everything that we heard about. Again, it's a really exciting morning here of discussion, and we're looking forward to our last session now. So thank you.